We're going to be continuing our conversation about Chapter 12, and specifically we are in Part 2, the nervous tissue. In this video, we're going to be focusing on the neuroglia. In our previous lecture, we talked about the neuron, and this is one type of cell that is located in nervous tissue. But there is a second type of cell that is located in nervous tissue, and this category of cells is called the neuroglia. In this video, we're going to introduce the general functions of neuroglia, and then we're going to look at the neuroglia that are present in the central nervous system and those that are present in the peripheral nervous system and understand their individual functions. So, what is the neuroglia? The neuroglia are going to be specialized cells that are going to act as scaffolding for the neurons. So they are going to wrap around and organize the neurons in particular arrangements. So it's going to hold them in place. And it also acts as insulators. I think this is one of the coolest things about neuroglia, and just kind of thinking about kind of the bioengineering, biomechanics of how the nervous system works. So what a lot of neuroglia do is they're actually going to wrap their plasma membrane, which is lipid base, around the neurons. And that lipid-based plasma membrane acts as a really good insulator. And so it's going to help control the electrical currents that are traveling through the neurons and make sure that they don't jump from one neuron to another. So it's going to make sure the electricity is going where it needs to go and not anywhere else, you know? So when we actually look at a neuron, almost the entire neuron is going to be covered by neuroglia to kind of contain the electrical current where it needs to be, with the exception of the synapses. The synapses are going to have to be exposed, so two neurons are capable of communicating with each other. Think of it almost as wires. There's going to be like rubber or plastic wrapping around wires so that when you have multiple wires kind of like in a bundle, the electricity won't jump from one to another. I know this really well. In college, a silly story is that my roommate really liked to pick up lamps and furniture off the side of the road, which is not a bad thing, but he never really checked if they were safe. And one time I was unplugging a lamp that my roommate found, and the wires were stripped. So the wires were exposed. I tried to unplug it, and it turned out that the electrical current jumped from the wire into myself, into my muscles, and I got electrocuted. That was not fun. So insulation is a really important thing when it comes to wires and when it comes to the nervous system. So that's a silly story. So this picture is not actually in your textbook, but I think it illustrates the neuroglia really nicely. There are going to be different types of neuroglia depending on where we are and whether or not we're in the peripheral nervous system versus the central nervous system. And we can see that neuroglia are going to have different shapes based on their functions, and they're going to be located and associated with different areas in like this particular picture is showing the central nervous system. They'll be associated with different areas in the central nervous system and in the peripheral nervous system. That's what I'm trying to say. Okay, so let's talk about the different types of neuroglia. So neuroglia is really a category of cells that are going to provide that scaffolding and insulation properties. And we're going to have different specific neuroglia in the central nervous system and in the peripheral nervous system. So right now what I'd like to do is go over the neuroglia that are specific to the central nervous system. The first one that I'd like to talk about are the astrocytes. Astrocytes get their name because astro refers to star and cyte meaning cell. So really it's a star cell. And if we look at the diagram, it does kind of have that star-like shape. It has lots of branches, and its branches will extend to the neurons, and it will wrap around the blood vessels that are feeding the central nervous system. The astrocytes will actually form the blood-brain barrier and actually control what comes into and out of the central nervous system, and limiting what can come in contact with the very sensitive nerves that are in our brain and spinal cord. Within physiology, astrocytes are being studied currently, and so we don't really understand all of the functions of the astrocytes, but some of the functions that we are familiar with are the following. They actually can regulate the amount of neurotransmitters in a synapse, especially in very high, in areas where there is very high neural activity. And it will do this by stimulating reuptake. What does that mean? What does reuptake mean? Reuptake is a way in which neurons can actually recycle neurotransmitters. So when a neurotransmitter is released from a neuron, what will happen is it will go into the synaptic cleft and it will bind to the receptors on the second neuron. But then what will happen is that neurotransmitter will be returned to the first neuron that was communicating, and the presynaptic neuron, and it can be used again. So it's a way of recycling the neurons, and astrocytes can help promote this recycling or this reuptake of neurotransmitters in highly active places in the brain. Another thing that it can do is it can be involved in signaling, increased blood flow through capillaries in areas where the brain is very active. So when the brain area is very active, those neurons are firing very rapidly, and we learned that neurons have a high metabolic rate. And so therefore, they're going to need a lot of energy, nutrients, and oxygen to be able to do its job. So when an area is firing very rapidly, these astrocytes can say, hey, let's redirect the blood to this particular area so that it can function the best it possibly can do. Another thing that it will do is it will control the ionic environment around the neurons, so it can control things like pH and all sorts of things. So that would be another example. And astrocytes are also important in helping form synapses during neural tissue development. So when the brain and spinal cord are forming in the embryo, not in the embryo, but in the fetus, what will happen is the astrocytes can help kind of regulate how synapses are being formed. So we could go and spend like an entire lecture on astrocytes and their functions. They're really amazing structures, and I kind of geek out about them. But what I would like you to know for this cell is that it is located in the central nervous system. It's the most abundant, and pick one of the known functions for the astrocytes to be able to know what it does. Okay, moving on from astrocytes. Astrocytes, and now on to microglia. Oh, microglia are super awesome. These are actually the least abundant cells that are found in the central nervous system. And really what they are is they're going to be the immune cells of the central nervous system. So their major job is to do what is called phagocytosis. Phago in Greek means to eat, and cyte means cell. And so phagocytosis is really the process of cell eating. So these microglia will roam the central nervous system and look for microorganisms that are infecting the central nervous system and kind of gobble them up and digest them. They will also look around for injured and dead neurons, and it will eat and kind of collect these neurons up. And so that they can keep everything nice and tidy and neat and free of infection. It is important to know that microglia, when we look at it from an embryological development kind of point of view, they do not originate from nervous tissue. Rather, they originate from blood. They are a blood cell. They're technically a white blood cell, and they're going to come from the stem cells that are located in red bone marrow, and then eventually kind of set up shop in the central nervous system. I actually did research on microglia when I was doing my rotations at U of R for graduate school. And we were studying the role of microglia in Alzheimer's. And what we had is we genetically modified mice to have Alzheimer's-like symptoms. And one of those symptoms is very active microglia. We don't know if the microglia causes these symptoms of Alzheimer's or if it is kind of a symptom in itself. But we were trying to identify ways of reducing microglia activation and therefore limit any destructive properties that they may have during Alzheimer's disease. So we actually genetically engineered a virus to create a protein that would limit the activation of the microglia, and we were trying to determine how effective this treatment was. Some of my role in this was to see if there was a significant decrease in microglia activation when we actually looked at the histology of these treated mice brains. And other members would actually do behavioral studies on the living mice that have been treated to see if they could remember to go through a water maze well. So just fun facts. I know it's a bit of a tangent, but there are cool things about microglia. The next type of cell that is in the central nervous system, that is a neuroglial cell, is called an epidermal cell. Epidermal cells are going to kind of line the hollow spaces in the brain and in the spinal cord. There are these hollow spaces in the brain called ventricles, and for the most part, they are filled with a fluid. This fluid is called cerebral spinal fluid, and the epidermal cells are responsible for creating and circulating cerebral spinal fluid. So what does cerebral spinal fluid do for the central nervous system? First and foremost, it actually fills the hollow spaces inside the brain and spinal cord, and kind of circulates this. This fluid is circulated through. So it fills those spaces, and this fluid also surrounds the brain and spinal cord, and it acts very much as like a cushion. As the brain and spinal cord are being moved around. So if there is a blow to the head, or blow to the spinal cord, the cerebral spinal fluid will actually cushion that blow. I kind of think of it like an airbag in a car. So like if there's like a car accident, and the airbag will deploy and it will kind of cushion the blow. The other thing it does is it circulates nutrients and removes waste. So cerebral spinal fluid is actually a filtered form of blood plasma that will have gone through the epidermal cells. And this it will allow for nutrients and other components to be delivered to the neurons that are found in the central nervous system, and it can help feed those neurons. Also, the neurons are going to produce waste, so the cerebral spinal fluid will pick up that waste, and eventually the cerebral spinal fluid will be sent back to the circulatory system and carried to areas of the body where that waste can be processed. So, in a nutshell, know that the epidermal cells are in the central nervous system, and they are going to deliver nutrients and remove waste. So choose a function that you'd like to remember for the epidermal cells. The last type of neuroglia that is in the central nervous system is called an oligodendrocyte. Note that there is the word dendro in this name again, dendro meaning branches and cyte meaning cell. And oligo means a few. So oligodendrocyte, when we actually translate the name, means a cell with a few branches, which makes sense. Really, if we look at the diagram here, we can see the cell has a cell body, and the cell will actually branch off and kind of wrap around different neurons and things like that. I kind of think it looks like an octopus. So it has a big body with lots of legs, and it's going to wrap its legs around neurons. So what is the function of the oligodendrocyte? I've already kind of referred to this a little bit. What's going to happen is it's going to make it's going to extend one of its arms out and wrap its arm tightly around the axon of a neuron. This is actually going to create an insulating layer called the myelin sheath. And the role of this myelin sheath is to speed up the propagation of an action potential. In the next video, we'll talk about why this occurs in a lot more detail. So just as a summary, the neuroglia of the central nervous system include astrocytes, the star-shaped cell, microglia, epidermal cells, and oligodendrocytes. Now let's go on to the neuroglia that are in the peripheral nervous system. The peripheral nervous system has two types of neuroglia, satellite cells and Schwann cells. Satellite cells, oh, I spelled Schwann wrong. Schwann has two N's, not one. My mistake. So satellite cells, their major job is to actually surround and isolate the cell body of the neurons that are in the peripheral nervous system, and it's going to control the local environment around that cell body. The Schwann cell, that's spelled with two N's, not one, is going to be, it's actually going to wrap around the axons and form the myelin sheath within the peripheral nervous system. So the Schwann cells and the oligodendrocytes have a similar function, but they're going to enact that function in a slightly different way. Oligodendrocytes are going to look like an octopus and are going to wrap its legs around many axons and kind of hold them together while it's forming the myelin sheath. While each Schwann cell is going to wrap around a specific area of the axon, and it's not going to have that kind of branching octopus-like structure. It looks almost like more like a rolled carpet around the axon. That's how I imagine it. So these are going to be the two neuroglia that are found in the peripheral nervous system, the satellite cells and the Schwann cells. And so what I'd like you to know for this is, where are these located? Central nervous system or peripheral nervous system? And what are their general functions? In the next video, we're going to look at how Schwann cells and oligodendrocytes form the myelin and learn how that myelin helps speed up the propagation of the action potential. Thank you for listening to this video, and I hope you have a wonderful day. Be good to yourselves. Bye-bye.